Hi everyone, we are group AT321 and today we are going to present on the topic BLAST, which is a basic local alignment search tool, which is an important topic in bioinformatics used for sequence alignment and analysis. My name is Prerna Dhakla and my group members are Sharon, Prerna, Edda and Tanvi. Together we have prepared this presentation to explain what BLAST is, how it works and its application in bioinformatics research. We hope this presentation will give you a clear understanding of the importance of BLAST and why it's considered a fundamental tool in biology data analysis. In the first slide, we have talked about one of the most powerful tools in bioinformatics, that is BLAST, which stands for Basic Local Alignment Search Tool. Now you might wonder why is BLAST such a big deal? With the rapid increase in DNA and protein sequencing, researchers needed a fast way to compare sequences. BLAST came into the picture in 1990s, introduced by Stephen et al. And his colleagues, and it has remained one of the most widely used tools ever since. And what makes it special is not just it's old and still relevant, it's that the BLAST completely changed how scientists identify tracks, diseases, and even study evolution. Today, we have walked through how BLAST works, its types, feature, application, and why mysterious e-value is important. Isn't just one program. It comes into five main types, depending on whether we are working with DNA or protein sequences. To make this less overwhelming, think of BLAST like a Google search engine, but instead of searching text on the internet, you are searching DNA and protein sequences. BLAST N, which is used for the DNA versus DNA, BLAST P, which is used for the protein versus protein, BLAST X is DNA translated into protein versus protein databases, and TB Blastin is for the protein versus DNA translated into protein, and TB Blastx is for the DNA translated versus DNA translated. So BLAST is like a translator and a detective. It can compare DNA with DNA, protein with protein, and even translate DNA to protein before searching.
Let's talk about how BLAST works. Now, how does BLAST actually work? Instead of comparing full sequences directly, which would take forever, it uses a clever shortcut. First, it breaks the query sequence into small words. Imagine dividing a book into little phrases instead of reading the whole book. These short phrases are called words. Second, BLAST looks for matches of those words in a huge database. Once it finds matches, it scores them using something called substitution matrices like PAM or BLOSSOM. Then comes the fun part. It extends the maths in both directions, like connecting puzzle pieces until the alignment score drops. The high scoring aligned segments are what we call HSPs, high scoring segment pairs. And finally, BLAST calculates the e-value, which tells us whether this match is meaningful or just random. We'll talk more about the e-value later. Let's talk about key features or characteristics of BLAST. So, why do scientists love BLAST so much? A few reasons stand out. It's fast, it can scan huge databases in seconds. It's versatile, that means it works with both DNA and proteins. It's sensitive, which is even tiny similarities are detected. It finds local alignments instead of forcing entire sequences to match, and it has a user-friendly interface that anyone can use. In short, BLAST is like the Google Maps of bioinformatics, quick, flexible, and super user-friendly. Let's talk about key features or characteristics of BLAST. So, why do scientists love BLAST so much? A few reasons stand out. It's fast, it can scan huge databases in seconds. It's versatile, that means it works with both DNA and proteins. It's sensitive, which is even tiny similarities are detected. It finds local alignments instead of forcing entire sequences to match, and it has a user-friendly interface that anyone can use. In short, BLAST is like the Google Maps of bioinformatics, quick, flexible, and super user-friendly.
The e-value or the expect value. So this e-value represents the probability that a sequence match is occurred by a random chance. So a lower equal e-value means that the match is more significant. So now let's demultiply the E value, um, which is for example, for example, if you blast a random sequence and you get a higher E value, it probably means that the match isn't meaningful. So but if the E value is very low, it will stay close to zero. It means that the match will be significant. So the rule is really simple. The lower the E value, the more trustworthy your match will be. This is why scientists mostly rely on E value while interpreting BLAST results. Now let's talk about the applications of BLAST. So what you can actually do with BLAST. So the applications would be endless, but here are some of the big ones that we use generally. So first one is the gene identification. It will find unknown genes by matching with known ones. And the second one would be functional annotations. It will predict gene and protein roles. And the third one would be evolutionary studies. It will trace relationship between one species and another and how species are actually related. And the fourth one would be medical research. It will detect pathogens and any kind of disease by studying and comparing DNA sequences. And the last one would be comparative genomics, which will compare genomes across different organisms. So next time when you hear about scientists discovering a new virus strain, chances are that BLAST played a very huge role in confirming what the organism which actually was. So to sum up, BLAST is not just a tool, it is a cornerstone of bioinformatics. It